Good afternoon, my name is Koichiro Majima, and today I'll be presenting our work on Spatiotemporal Variational Autoencoder for modeling continuous tissue dynamics, a project developed with my boss Teppei Shimamura at the Institute of Science Tokyo. To begin, let's consider the biological context, morphogenesis. The process of tissue formation is guided by signaling molecule called morphogens. These morphogens form concentration gradient, and a cell position within these gradients directly determine its gene expression and subsequent morphogenetic fate. Therefore, we hypothesize that gene expression is a primary driver of morphogenesis. Modern tools like spatial transcriptomics allows us to capture gene expression with spatial information at an incredible resolution. However, these static snapshots for disruptive measurement. A major challenge is to understand the continuous process of development. Current computational methods such as MOSCOT and STBCR often use optimal transport to map cells between these time points. While powerful, these methods can be limited in capturing the non-linear and complex continuous dynamics of tissue growth. To address these challenges, we set out with three specific goals. First, we wanted to create a model that could capture the continuous dynamics. Second, we aimed to learn both the pixel state and its spatio-temporal evolution simultaneously using a unified VAE and neural ODE framework. Finally, our goal was to enable predictive simulations, allowing us to infer past or future state and identify the key genes driving these processes. This slide provides a conceptual overview of our model and its applications. The top panel illustrates our core concept. We model pixel dynamics using a learnable potential energy landscape. You can see how pixels, represented as spheres, move and change state over time by following the changes in the landscape. The bottom panel highlights the model's practical uses. We can predict gene expression and morphology at unobserved time points, infer cell movement to understand how individual pixels contribute to the large structure, extract the key gene that regulates and drive these morphological changes. This slide breaks down our model's key component, each indexed by time t. X, the input data, observed gene expression for each pixel. X hat, the model's output, the reconstructed gene expression data. Z, a simplified summary of the expression data compressed by a VAE. S, the pixel's location. U, a learnable energy landscape that guides how and where pixels move. This is the graphical model of our proposed method. First, we use a VAE encoder to compress spatial gene expression data X into a low-dimensional latent space Z. This process captures non-linear gene expression relationship by inferring the variational distribution QZ given X. We define a unified state vector for each pixel Y by combining its latent gene expression Z and its spatial coordinate S. The dynamics of this unified state are governed by a learnable potential function U. This function is a neural network that defines a time-dependent energy landscape over the combined spatial and latent space. According to the equation, U sub I, t equals f of y sub I, t, t. The shape of this landscape is learned during training. We model the change in a pixel state over time using ordinary differential equation, ODE. The pixel's velocity, or dtdy, is determined by a neural network, G. Critically, this function takes the gradient of the potential, nabla u, as a direct input. This design ensures that the pixel's movement is physically guided by the energy landscape. By chaining together the single OD steps, we can generate a complete path that connects all of the time points. By formulating development with an ODE, we treat it as a truly continuous process. This continuous formulation is powerful because it allows us to query the model for a pixel state y equals the vector z, s at any arbitrary time. This let us generate a smooth movie-like interpolation of the entire dynamic process. Once we simulate a pixel's trajectory to get its state y, we use a VA decoder to reconstruct its gene expression profile. Our model is trained by minimizing the sliced Wasserstein distance, SWD, between the predicted and observed pixel distribution at each time point. This process adjusts the potential U and ODG network, shaping the energy landscape to ensure our simulated trajectories statistically match the real data. From here, I will explain the mathematical background of proposed model. We use a VAE to run a simplified representation of the complex gene expression data. It works in two steps. An encoder compresses the original data into a simple low-dimensional summary called the latent space Z. Second, decoder then uses that simple summary to reconstruct the original data x hat. Having established a low-dimensional latent space, we define a unified state vector for each pixel, denoted as y. The core of our model is learnable potential field U, which is neural network that defines a time-dependent energy landscape. The evolution of a pixel's state is then governed by an ODE. As shown in the final equation, a second neural network determines the pixel's velocity dt dy based on its current state y, the gradient of potential nabla u, and time t. By solving this ODE, we can trace a continuous trajectory for any pixel, allowing us to predict its state at any point in time. Our loss function is weighted sum of three key components, as shown in the main equation. The most important component of our loss function is sliced Wasserstein distance. Since we compare entire population of pixels rather than one-to-one pairings, we need two metrics that can measure the distance between two distributions. The SWD achieves this by projecting the multi-dimensional pixel state onto many random one-dimensional lines and then calculating the average cost of transforming one distribution into the other along these lines. This loss ensures that the spatial and transcriptomic structure of our simulated pixel population becomes statistically indistinguishable from the observed data. To test our model, we created a toy dataset that simulates dynamic nonlinear tissue growth. We then tasked our model and two other methods, linear interpolation and optimized transport, with predicting an intermediate state of this growth, giving only the start and end points. The top row compares the 2D spatial predictions to the ground truth. The bar chart at the bottom provides gene expression error and spatial coordinate error. Our model achieves the lowest error in both charts, demonstrating superior accuracy in predicting both the gene expression, and the spatial arrangement of pixels. The data used in this study was sourced from the MOSTA dataset, which was generated using StereoSeq. We utilized a specific subset of this atlas, focusing on such subsection from mouse embryos at 3 consecutive developmental stages. We simulated continuous tissue development by running our learned ODE. The animation shows the latent space Z on the left and the corresponding cell type prediction on the right. The resulting simulation is spatially coherent and biologically plausible, confirming our model successfully learned the key dynamics of development. This slide displays a snapshot of the simulated developmental processes, with cells colored by type and arranged chronologically. The simulation generates structures that are remarkably consistent with non-embryological events. For example, the emergence and organization of somites is consistent with biological facts. This stacked bar chart provides a quantitative summary of our simulation, showing how proportion of different cell types change over the developmental timeline. The chart correctly captures organogenesis dynamics. For example, it shows the brachial arch jaw structures are prominent early on. As development progresses, the relative proportion of the brain and the surface ectoderm increase significantly, reflecting known patterns of growth. Besides, our model accurately predicts gene expression. This heat map shows our simulated cell types have the correct marker genes, such as FEZ1 in the brain and PCSK9 in the liver. This confirms the biological accuracy of our simulation. On this slide, I'll explain our model is not just a predictive tool, but also a powerful analytical engine for studying tissue development. First, our model learns a continuous vector field F that governs how pixels behave. As the equation shows, this field defines the velocity for our pixel's complete state Y, which includes both its gene expression and its spatial position. By calculating the divergence of the spatial velocity, we can measure local pixel flux. In other words, where the tissue is expanding. This heat map quantifies tissue dynamics, showing the average absolute divergence for each cell type over time. Brighter colors highlight regions undergoing significant expansion or contraction, such as the liver and heart, while darker colors represent more structurally stable regions. This observation aligns with established developmental knowledge. For example, the high divergence we see reflects major biological events known to occur around the embryonic day 10, including the differentiation of endothelial cell in the heart and the significant development of the brain. Our model can identify genes that drive cell movement during tissue formation. This bar chart displays the top genes most predictive of this movement, ranked by correlating their expression at one time point with the distance a pixel travels to the next time point. Notably, the identified genes are biologically relevant to morphogenesis. For example, the list includes cytoskeletal proteins, for example, actin-1 and actin-2, and their regulators, NHS, which provides the physical machinery for movement. In summary, our key contribution is novel framework that learns a continuous developmental vector field from discrete snapshots. By unifying gene expression and spatial data with a potential guided neural ODE, we successfully simulated mouse organogenesis and generated spatially coherent tissue structures. This powerful approach enabled the quantitative analysis of morphogenesis, allowing us to identify regions of achieved growth and their underlying genetic drivers. Thank you for listening.
Thank you very much. Do we have any questions? Yeah.
Thanks a lot for the talk. I had a question. How do you handle the fact, do you register the slices across time points? Because I don't see how in absolute values, the spatial coordinates, they could have a meaning. Like if you were to translate a slide or rotate it, it would not change anything to the tissue. But if you use the raw spatial coordinates, it would change everything in the analytical analysis. So I wonder how you handle them.
That's very difficult point. And actually, I'm doing that manually to show same direction.
So you're manually registering the slides, like rotating.
Yeah.
They have the same orientation.
Yes, that's right.
Do we have other questions? Okay, if not, let's thank again our speaker.